So much for having me. It's fantastic to be here, and I'm delighted to be able to talk to you about this very, very sensitive subject, because partnerships, let's face it, are the center of the universe. So partnerships, I'm going to start off by talking about the amazing thing that is coral reefs and the partnership that this particular ecosystem makes with humans. So they are in of themselves just spectacularly beautiful places, and I was enamored by them very early on in my life, and really it was because they were a convening place. They were the cities of the underwater world where you find hundreds and thousands of other species that convene in the spaces that they build. But those spaces, those structures that they build are really not academic at all. They are the natural breakwaters, so they protect the land masses that they surround. So here in Hawaii, you see these fabulous waves breaking off the barrier reefs, and that is where the energy of storm waves is being broken and protecting the land and preventing the erosion of the land masses behind them. The things that live in them and on them are worth millions of dollars. They feed 500 million to 1 billion people. In some Pacific islands, the reefs are home to fisheries that support 70% of the protein eaten in those places. 70%. That's an enormous amount of food. And here in Hawaii, you can't really talk about the reef without talking about tourism. People come here because Hawaii is spectacularly beautiful, both above and below the water. And that natural beauty is the economic driver of the state. So the coral reefs are absolutely fundamental to the economic health of many, many islands, including Hawaii. But really, when we talk about coral reefs today, we tend to talk about how spectacular they are. And then in the next breath, we tell you that they're going away, and that they're going away at an alarming rate. And really, this reflects the fact that coral reefs live very, very, very close to the temperature at which they can survive. So they are right on the edge. And as our planet changes, as the waters become warmer, that threshold of them being healthy to unhealthy is being crossed. And it's being crossed ever more frequently by excursions in the ocean temperature that are pushing corals along well beyond their limits. And so we call this kind of degradation an ecological disaster. It's an ecological disaster to think that in our lifetime, we might wipe out a system that is so fundamentally linked to humans. It just makes no sense to me when I think about it. I've been studying coral reefs. I did my PhD in 1990. And I did my PhD on the North Shore of Jamaica. And some of those reefs were some of the first to show really, really serious declines. And this was sort of the perfect storm of, first of all, overfishing, hand-to-mouth fishing to feed people's families, coupled now with massive storm damage and a disease outbreak of the thing that ate the algae that was overgrowing the coral. And so we saw these thriving reefs in the Caribbean become graveyards over a period of a couple of years. Massive temperature disturbances have occurred. There have been 3 global bleaching events, and the last one was in 2015 and 2016, continuing today. We call them global bleaching events and global disturbances on corals because they affect a huge amount of the globe. So that means the temperatures around the globe universally went above the threshold last year, at which a lot of corals could die. And many of you will have heard of the terrible devastation on the Great Barrier Reef, the northern portion of the Great Barrier Reef, where the corals, 22% of the corals on the Great Barrier Reef died last year. I head up the society, that is, the learned society, all scientists who work on coral reefs, and we put out a consensus statement saying that if we do not change the way we do business, all corals and reefs on the planet will be massively degraded by 2050. 2050. So I'm just going to let that sink in a minute. 2050. That is around the corner. We don't have time. So understanding what is going on, getting a handle on what the things that are driving these declines at such alarming rates is a critically important area of study. And University of Hawaii has an enormously dynamic group of people who are working in this area. So before I go on to talk about some of the science, I just want to talk about the special creatures that we are really talking about here. And the very thing that drew me to corals in the first place wasn't all of this discussion of them dying. It was this extraordinary beast, this tiny, tiny ancient animal that partners with a tiny plant that lives inside of its tissues, pausing for a moment to think about what that means. Okay, this is an animal with a plant living inside of its cells. That's crazy talk. No, we can't do that. There's just no way that we would tolerate having something as foreign inside of our tissues. You know, whenever we get a foreign body inside of our tissues, a virus or a bacteria, we do our best to try and get rid of it. But corals have evolved over millions of years in tight partnership with these tiny plant cells that give them their colors, these dark brown colors that you see on the reef, is because their cells contain millions of these tiny algae. So that in this image on the bottom left here, you see these bright red dots. Those are the tiny plant cells inside of a single polyp of a coral. This polyp is only 3 millimeters across. You can see how many of those red dots there are. And this green here is a bright green fluorescent protein that's inside the coral animal itself. No dyes were used in the generation of this image. We just illuminated it with an ultraviolet light, and this is what they look like. These tiny, tiny polyps combine by the thousands to create individual coral colonies, and many colonies assemble on a reef to create a coral reef that whose collective effort can be seen from space. These structures that are created by these tiny ancient organisms can be seen from space. The partnership between the algae, these tiny little plant cells inside the animal, and the animal itself enables the corals to lay down the bone that creates that reef. So they literally grow up over this bone-like structure that they secrete. And it's that bone-like structure, of course, that is the hard barrier to storm waves. So it's the union of these two very unlikely partners, a plant and an animal, to create these extraordinary structures that support billions of dollars of revenue. When you go out on the reef, what you see is many different shapes, and the shapes all intertwine with each other. Different coral species grow in different growth forms, and we don't really understand why that is. But actually some of the newest work in my group is asking the question of, does the actual nature of the partnership create differences in the structures that they create together? So are nuances in interaction between these tiny algal cells and these animals resulting in structures that are very different among different types of partners? So that is a really exciting thing. But when these plants are inside the animal, they do what all plants do. They do what is so critical to our health in terms of the production of oxygen. They photosynthesize. They're able to take the gaseous carbon dioxide that we breathe out and use light energy to combine it with water to create an oxygen molecule. They give off the oxygen, they give that small sugar to the animal, and the animal can feed from within. It has a food factory living inside of itself. I mean, how amazingly cool is that? Imagine world hunger would be solved if we could only do what these corals can do. So they're amazing, amazing creatures. They have been on the planet, the corals themselves have been on the planet for over 200 million years. And remember, I just said we could wipe them out by 2050. In our lifetime, within 75 years, we will have taken a system that has thrived on planet Earth and destroyed it in our lifetime. And so what happens when corals get stressed? What do they do? What do they look like? What is the manifestation? Well, remember I said these tiny alpha cells are inside their tissues, and it is the color of those cells that makes corals look dark brown. So when you see a dark brown coral on the reef, you know, it's healthy. It's looking good. As corals get stressed, they manifest their stress in a really bizarre way. They essentially no longer communicate with the plants that are living inside them, that feed them. They see them as a foreign body, and they get rid of them as quickly as they can. And we know from many studies that that reflects a breakdown in the capacity of the tiny cells to photosynthesize, to use the carbon dioxide and water to produce food. When they're no longer producing food for the animal host, there's something in the host that recognizes that something is awry. And at that moment, they say, it's better not to have this thing inside of me any longer. I'm spitting that thing out. And so what you see is these dark brown corals go pale as they lose the millions of cells that they contain. They contain about 1 million algal cells per centimeter squared of tissue. That's a hell of a lot of cells. So they lose the dark brown algae, they turn gradually paler and paler until they go stark white. And when they go stark white, they are now devoid of their food supply. So you can imagine they're now starting to feel a little unwell. They can no longer really have the energy to grow. They start to calcify less. And there's really a bit of a race against time. If the conditions that cause the stress, that white coral can turn dark brown again, and it can recover. But if the conditions persist, often a white coral will die. And a fully bleached individual, like the one there on the far right, usually means that that coral will die. And when you see a whole reef that turns white before your very eyes, a large portion of that reef will die. Is high. Most of the time, when we're talking about mass coral bleaching, we're really talking about temperature. Other things that can cause corals to bleach, and we know that many pollutants will elicit the same stress response by a slightly different mechanism. We know that sediment stress, when you've got a lot of particulates in the water associated potentially with runoff from land, it can also cause the coral to turn white. So measure differences in the health of the coral that relate to the other change or ocean change stressor, which is ocean acidification. A change in the chemistry of the water will also induce a shift in the health of the coral that will manifest as coral bleaching. So it is a ubiquitous stress response, much like a fever is a ubiquitous stress response in humans. But what people don't often talk about, and what I'm going to focus on today, because I have to say I'm a cup half full kind of gal, so I can tell the tale of the things that die, but I actually prefer to think about the things that are surviving, because it's the things that are surviving that will frame whether or not we have a reef tomorrow. So the little known secret about corals and coral bleaching is that not all corals will manifest the stress response equally. So just like in this room, if we all got infected by some infectious agent, some people would get very sick and others would not. And it's the ones that don't that I'm most interested in. And what we see is that at the scale of an individual even, we can see variation in the color patterns of a coral. The top surface of this coral is very bleached, the underside is not. Unhealthy, healthy. These two species, side by side, one species is stark white, the one is still brown next door to it. Different species have different tolerances for stress. And even on a reef, you might see individuals within the same species, some of which are doing better than others, some of which are completely brown, and the one right next door completely white. And we know, at the scale of the entire Hawaiian archipelago, that during bleaching events, some reefs are really badly hit and other reefs are not. And the question is, why? Why? Well, that question has actually been my entire career. And I'd like to say that I have no idea. No, I have some idea, but I don't understand them more as well as I would like to. Because I think what's so fascinating about these organisms is they've been on the planet for 200 million years. They have tricks up their sleeves that perhaps we have not even begun to explore. There is so much new biology to be found. And when I first studied them, I studied them because they embody the biological complexity that you find everywhere else in the animal kingdom is found in the corals themselves. And so I thought, what a great model system. I'm just going to study these things because they're so complex, I'll never understand them. And I was completely right. But what we do know is that there are several things that we can look at that explain some of the differences in the way they respond to stress. And there are three things really that come into play. First of all, these really intimate partnerships between the algae and the animals themselves. You know, when we first discovered these partnerships, we thought there was a single species of algae that was in symbiosis or intimate association with all coral species on the planet. And then we learned, using new tools, molecular tools, that even though all of the cells seem to look very similar, when we really look at how different they are at the molecular level, at the DNA level, the very characteristic of the species, we found there were literally hundreds of different types of algae. And who you partner with is critically important. So some of the species are extremely narrow in their functional range. And what I mean by that is the temperature range under which they can function well inside an animal is very, very narrow. And there are others that have much wider range, that they can withstand much higher temperatures. We call those thermal tolerance. And the thermal tolerance, obviously, if you're coming into summer months when the temperature gets very hot, it's really good news if you have a thermal tolerant inside of you because they keep functioning at much higher level, a much higher temperature. So the reality is that this partnership choice is critically important. Now, do you get to choose? That's the question. And for some species, the answer to that is no. Your parents give you the type of symbiont that you're going to have for life. So what you get in the egg from your parent, your mother, is what you have. But for 85% of coral species, they have to reinvigorate these symbiotic relationships every generation. And so in that moment, there is an opportunity for them to choose a type that might be optimally suited to the future or not. Actually, what they do is they are selecting symbionts that are available to them. And it's very rare for them to find these thermally tolerant symbionts. Because what we know now is that these thermally tolerant symbionts are great when the temperature is very high, but they don't feed their animal host as well. So the long-term prognosis for an association with a thermally tolerant symbiont is you get through a bleaching event, you may survive, but you may never reproduce again because you don't have enough energy, because this particular symbiont, we now believe, is an opportunist. It's basically coming into habitat that is available to it because the host is less defended. It's a really, really interesting nuance in this interaction. So the first thing that's important in survivorship is who you partner with. I just told you about one partnership, but we now know that corals are covered in bacteria, and they have many, many, many bacteria living inside their tissues. And who those microbial partners are also matters. So this is a revolution in biology going on right now. The importance of small things on the behavior and the performance of large things. And there's nothing more profound than this study in humans that shows that our sleep cycles are completely dictated by the healthy bacteria that live in our gut. The way that we assimilate our diet, the way that we use nutrition, is completely controlled by the healthy bacteria that live in our gut. This is an astonishing level of interaction between large things and small things. And some would argue that it is as important to our understanding of biology today, a new synthesis, as important as Darwin's evolution. It's massive. And so this shift is all to do with the tools that we now have at our fingertips to explore what's going on in the micro realm. We can visualize things in that realm that we could never, ever visualize before. And for me as a biologist, I have to say, it's this level of new discovery that kind of unfolds when time passes and new tools come to our table that allows us to think about the fact that there may be many, many aspects of biology that we have yet to discover. Consciousness, what sets character, altruism, narcissism, all of these things, we have zero understanding of how those things are framed in our own species. So how do we hope to understand them in a species that can't speak? So, who you partner with, microbes, macro eukaryotes, the algae, really, really matters in how you deal with stress. But what seems to be critically important, as is critically important in humans, is who you partner with when you choose to reproduce. Who you choose to partner with when you choose to reproduce. We know the base genetics of this system are incredibly complex and incredibly important. So, the eggs and sperm from two parents who have lived in stressful conditions and survived it will produce offspring that seem to have a better opportunity to survive stress in their generation. So it's critically important. Partner choice is everything when it comes to reproduction in corals. And of course, it's a very stochastic event, right? Corals reproduce on 1 or 2 months a year, 1 or 2 days at a specific time of night. This marvelous event happens when all of the eggs and sperms are released and they mix in the water column, and it's crazy who crosses with who crosses with who. It's fabulous. It's nature at its finest, really. It's really, if you've ever been close to a coral reproductive event, it changes your life. It really does. It's just an astonishing feat of nature. But there's one other thing that seems to be really important, and it's really, to me, in terms of stress survivorship, it's sort of implicit. If you've actually survived stress once, right? If you've bleached and regained your color and you go on to grow, it seems like your opportunity to face a future stress is better. So, along the lines of what doesn't kill you makes you stronger, right? So we understand that intrinsically for ourselves. We understand that what we experience. So I always laugh and say, I use the analogy, the first time I stood up and put a microphone to my mouth to give a talk, I was so scared. People in the audience could see me do this. It was terrifying, right? Well, the second time I did it, it was less terrifying. And now I do it and I love speaking. It's like an everyday thing for me. It's just because I'm used to it. It's within my experience envelope. I have completely inured to that event. And corals seem to do the same thing. If they experience something and survive it, a stress event, they seem better able to face the future. Now, to me, that's a very hopeful thing, right? We've identified 3 things that seem to be fundamental to how corals will survive the future. Well, I'm a coral reef scientist, and so I report all of this to my friends. And they all happen to be scientists. And I do this through one of the most inaccessible mechanisms known to man or womankind, the peer-reviewed scientific literature. Right? I mean, I work for hours to put these papers together, and they go through all this peer review. People say it's not very good work, blah, blah, blah. But at the end of the day, we have all this information. We have all this information about how our reefs are declining and these mechanisms by which corals are either dying or not dying exist, and we always conclude in our studies. And this work is directly relevant to the management and conservation of coral reefs. Well, I hate to be blunt. I've written that 1,000 times. How is a question I had to ask myself. And in fact, in 2010, I went on sabbatical and I actually asked myself that question. Really? Is your work really of direct relevance? To who? How? Well, that's the challenge, isn't it? We have a reef that is declining, or reefs globally that are declining at a rate of knots. We have science that's compartmentalized in the ivory tower. We choose not to speak about it because we might not be 95% confident about what the outcome is. And at the end of the day, our science is disconnected from the very system that we would hope to be able to protect. And so the challenge then is, how do we connect our science to this bigger issue of climate change and the death of coral reefs? How do we use our science and harness that information to make a difference, to attempt to change the trajectory? Can we? And I would argue that the answer is yes, because we have to focus on the survivors. We can't be just reporting death all the time. Right? Everybody's getting exhausted. Oh my God, there's another newspaper article about coral reefs dying. We may as well give up on the system. What's the point of doing anything to protect them? They're dying. We should just move on to the next cause that's important, which is really something that upsets me, because actually I think there's a lot that we can do about it. So the question is, how do we harness this basic knowledge that genetics, who you partner with, and your past experience is important in whether or not you survive climate change? Well, we assist the system. That's my conclusion, is we ask whether or not we can actually accelerate the naturally occurring biology to keep pace with the changes that we're seeing on reefs, to develop corals, potentially, that can withstand climate change. Because we know that the real problem is that the intrinsic rates of change in the environment today, temperature changes, ocean acidification, pollution, are outpacing the intrinsic system, the intrinsic biology that maintains the system. And we know this because, conservatively, we have lost between 40 and 50% of the world's reefs already. If the natural system could keep up and could replenish, we would not have lost 40 to 50%. It's as simple as that. It's just an equation, right? Something in equals something out equals zero. When something out of the system outweighs the something in, you go into the negative. And that's where we're at. We're at the negative. And so I told you, we think the things that dictate whether or not corals survive are who they partner with, the genetics of their parents, and their experience. Well, why would we not then try to emulate those things, control those things, direct those things? And so my group, along with my colleagues in Australia, put in a fairly radical proposal in response to an international competition for ideas. What do we do? And our ideas to assist evolution won that competition, and won us a fairly large chunk of change to do the proof of concept that we could selectively breed corals. We could control the parentage. We could try and take the strongest of the strong from the reef, take their eggs and sperm, and deliberately cross those to produce the strongest individual. Now, selective breeding is not radical by any stretch of the imagination. We've been doing it with almost everything that we eat for as long as we can remember. All of our domesticated animals, you name it, we've selectively bred it. We have directed the evolution of almost all species that we deem to be economically important to us. So on land, this is a completely benign idea. But in wild systems, this is a very radical idea, right? This is like, we have never had to selectively breed a wild organism before. But we do that, don't we? Because when we think about endangered species, we've waited too long for those species. And so we are okay when there's only 4 left on the planet to cross cousins. We do it all the time in zoos. And we do it usually in vitro because the success of that cross will be so much greater when we do it. We've waited too long, and I keep saying, let's not wait until we've got 4 corals left on the reef to decide to do something. Let's think about what we need to be doing now. So when we intervene, we can do a lower risk intervention. We don't have to do radical crossing. We have a huge amount of biology left. Let's store it, let's biobank it, and then let's selectively breed it. Or maybe what we should do is modify the symbionts. You know, you've probably heard that people are doing fecal transplants in humans. So there was this amazing twin study that was done, where there were 2 twins, one of whom was obese and one who was skinny. Of course, their genetics are identical because they're twins. And they took the fecal matter from those 2 twins and they transplanted that into mice, and then they fed the mice the same diet. And the one that got the fecal transplant from the obese twin the mice got obese, and the one from the skinny twin stayed skinny, same diet. Right? And then they crossed the mice fecal transplants, and they modified and changed that relationship. So at some level, we know that you can intervene by changing microbes and have a very different outcome. And so why would you not modify the symbiosis or the microbial communities to effect a community that has the best chance of withstanding future conditions, warmer conditions? Why not? And if we know that experience and surviving experience actually gives you a leg up, why not give corals experiences that they survive? Why not ask the question of how high temperature for how long can a coral survive easily? And if they survive it, does that give them the ability to face a future stress more easily? And so we are doing that proof of concept. We are on Coconut Island now beginning to do these challenges. And this project is often called the Super Coral Project, because at one point I analogized the whole thing to what we do with super athletes. What you do is you go out into the population and you pick the very best performers. I'm looking for the best footballer, right? We have scouts who do that. We bring those footballers into the gym and we train them. Well, I've gone out, I found my really high-performing corals. That is the corals that don't seem to bleach when all of those around them do. I bring them into my lab, into my gym, my aquarium gym, and I'm conditioning them for the future. That is, I'm running them on my environmental treadmills. They are doing pretty well. And when we bring our athletes in, we train them on a treadmill and then we give them great nutrition. Well, remember, these symbionts feed the animal. So what I do then is ask, can we modify the symbiosis to give them the very best nutrition possible so they actually are fat enough to lose weight during a stress event? Because we know that the fatter the coral is going into a stress event, the more likely it is to survive. So it's called the Super Coral Project because essentially I'm selecting the super athlete corals, I'm running them on my environmental treadmills, I'm modifying their symbiosis, and then I'm looking at whether or not the next generation is doing better, worse, or the same. Right? All I've done is taken the science that I've been doing essentially for 25 years. I know I look like I'm only 10, but I've been doing my science for 25 years, and I've taken it one step further. I've taken it from my academic pursuit, observing and reporting, and I stepped out of the box and said, now I'm going to use it. And I, as a fairly senior coral reef scientist, am going to be the one who risks my career and my reputation to step out and say, it's time. It's time that we change the way that we, as scientists, are doing business. It's time that we think explicitly about how our science serves a larger agenda, not just of our own interests, right? So I'll admit quite freely, I got into the system because I was fascinated by the biology and the tools that were available to me to explore it. And the creativeness of an academic lifestyle is unbelievable. right? You don't really get told what to do for a living. You design things, you do things, you teach people. It's a phenomenal career. But at this point, as a scientist who spent 25 years looking at my system now start to die before my very eyes, I feel like it's my responsibility to take my skills and apply it to a solution. And it comes to this perspective now, we as scientists have an enormous amount to offer in the solution space. But our reward system doesn't necessarily reward us for stepping into that space. At my point, it doesn't really matter. But for a junior career person, it's a little bit of a more risky move. So, all of these shifts in the biology, all of these tools, all aimed at changing the way the corals are actually performing by manipulating the base genetics, the symbiosis, or the experience history. That's all that we're doing. It's not genetic engineering. We're not genetic engineering. We've genetic engineered just about everything. We're not doing CRISPR on the system, which is gene editing to effect a change that we think will be good. All we're doing is accelerating what nature does. Well, this work was put out into my broader audience, and I have to say that I took an enormous amount of criticism for doing this, right? And every single one of these criticisms is valid or not valid. But every single one of them pointed me in the direction of a problem. People are so scared to act. People would say, but what if, but what if, but what if, about everything that we were putting forward. What if we plant, I know, the next cane toad? Well, they would say, but we're not working on one system, we're working on all of the coral species that exist here. We're doing it all in the laboratory, so there's no risk of releasing anything in the wild right now. We're in a proof of concept phase. What if we get genetic narrowing when we selectively breed, too much? Well, we've been doing selective breeding for a very long time as humans, so we know a lot about how to prevent genetic narrowing from other systems. We can take all of that information, we can use that all, and we can really be cognizant of that going into the problem, not at the other end. We're at the beginning, not at the end. We haven't got a fait accompli. We don't even know whether this will work. So criticizing it based on these what-ifs seems a little bit over the top.
What is cane toad?
A cane toad was an introduced toad in Australia that literally killed everything around it and grew to be a huge pest. And, to be honest with you, there's been many instances of us doing things like that as humans, but it wasn't biologists who introduced that. I would say there are most instances where a biological control was introduced into a situation. It was introduced by an agriculture person, or the Army Corps of Engineers, or people disconnected from the actual behavior, knowledge about the behavior of the system. And so this is a critical thing, actually. This question is so good because we're biologists, and our motivation and our framing and foundation is over 50 years of basic science and 25 years that has led us to our conclusions about genetics. My experience, this is a huge amount of evidence that supports this agenda. But what we're really up against is fear. We're really up against, within our own scientific community, fear that science and scientists are acting in an unusual way by taking science to solutions. But I think fear that we as scientists are observing our system die, and that it is going to die before our very eyes if we do nothing. And most people feel paralyzed against the enormity of that statement. And I would argue, well, what if we get genetic narrowing? It could happen. We'd be very cognizant to avoid it, but what if we do nothing? What if we do nothing? At this moment in time, what if we choose to use all of these what-ifs as the reason to stop this kind of work? what will happen?
We'll get genetic narrowing anyway.
We'll get genetic narrowing anyway, because nature, climate change is doing the most uncontrolled experiment in genetic narrowing. We just call it reductions in biodiversity. People talk about biodiversity, species being wiped out before we've even discovered them. Right? Well, that's just genetic narrowing. But at a macro scale, we're talking about genetic narrowing in a single species, but that species may not even persist through the next decade. So I would argue, let's not what if we do, let's talk about what if we don't. What if we don't do something now? Now, most of my colleagues would say what we should do is we should put enormous pressure on our government to mitigate the drivers of climate change. Whether you agree or not that fossil fuels and fossil fuel burning is causing climate change, the reality is the evidence is completely supportive of fossil fuel burning actually being the driver for climate change. There is not a single scientist that I know who would think there was any doubt in that argument whatsoever. I agree that we should try everything we can to stop the train wreck that is climate change. But we don't seem to be doing enough. And even if we change the way we do business and we limit warming to 2 degrees, which is really the best case scenario at this time, it's too much for corals. It's too warm. So there's no choice at this point. We have to think outside the box about, do we want some corals in the future or no corals in the future? Is this set of solutions going to be enough to protect corals worldwide? No. But will we still have corals in the wild if we act now and we are able to engage many people in many places in these kinds of efforts? The answer is yes. Because frankly, I can't sit back and wait anymore. I can't sit back with the knowledge that if I do not apply my skill set, that it is likely that the only place that I will see corals left in the world will be in an aquarium. And if that comes to pass, we will have a situation that is so dire in terms of ecological refugees and starving people that all of the biology, the biodiversity discussion falls away, because now it comes back to the relationship between corals and humans. It is so interwoven in parts of this world that to have no corals is to completely modify the way that people are able to live in their spaces. We already see the implications of death of reefs and increasing temperature and also sea level rise. We are all seeing the first ecological refugees moving from places that are no longer inhabitable in the Pacific. It is happening now. And if all corals die by 2050, it is going to be a disaster in terms of people, because let's face it, there are going to be more people on the planet than there are right now.
Let's see. I think we all probably are aware that there have been previous extinctions that have affected coral species. We've seen evolution and change and so forth. So I think I agree most generally with what you're saying. I'm very worried too. But I'm thinking that I would disagree with your prediction that there are going to be 0 species of corals around by 2050. I think there will be a lot fewer, but I'm thinking that why would we say there would be 0? Are we saying that we're going to narrow down to such a few and then some sort of meteorological event will just break the
Well, so I think your point is really an interesting one. So historically, there have been extinctions on geologic time
even
record.
prehistoric?
Yeah, I mean, but today, in modern reefs, there has never been the rate of change or the rate of loss on reefs ever in history. Ever.
So you're saying the rate of evolution is much slower than?
Absolutely. And because they're very long-lived animals, so they cannot replenish their populations quickly enough to essentially keep up with the increasing time period, the narrowing time period in the temperature that is pushing them outside their comfort zone. So what's happening is, it's like the first time a coral gets bleached, if it survives it, it's great. But if it doesn't reproduce, and then it gets to another bleaching event, it will actually eventually just die. It will die, and there will be no propagules to replenish the reef. And actually, the perpetuation of coral reefs. So remember at the beginning I said, all coral reefs will be massively degraded. Now, does that mean that there will be no single corals left on the planet? No, there'll be one or two parties. But they won't form reefs, because reefs require an assembly of species, and they require an assembly of different age classes to create spaces that actually support the life that supports people. So, in answer to your question, it's the rates of change of the environment right now that have never been so steep that associate with climate change, and the number of stressors that are in the nearshore environment that relate to our activities on land. So because coral reefs, for the most part, are immediately adjacent to land. Our pollutants, right now we're doing a study on caffeine. What is the effect of caffeine or birth control pills that we're flushing into the nearshore waters that are now disrupting all of the reproduction of the corals? Sunscreens, as you know, very big here in Marama Bay. What is the effect of the chemicals in sunscreens? The sediments. All of these things are coming together and coalescing with temperature and acidification. It's like, we're seeing it play out in places like Beijing in humans, where the air pollution is so bad that people are getting massive chest infections, can no longer live because of this. It's the same problem. It's compounding one thing on top of the other, and the base rate is changing, but it's the excursions in the summer that are getting higher and higher, and are already pushing. So where we might have expected a bleaching event maybe every 6 or 7 years, we're now predicting bleaching events every 2.
So maybe to summarize, and then I'll just interrupt and more about it's not only the increase in temperature and the speed at which the increase in temperature is occurring, but it's other anthropomorphic
anthropogenic stressors,
effects,
yes.
stressors.
It is, climate change is an anthropogenic stress. I think that's the thing, to not recognize the relationship with fossil fuel burning as a stressor. It's just another thing that we do that leads to a set of environmental conditions that do not promote coral growth. And so, I'm going to be blunt. 2,000 coral reef scientists agree on this thing. Experts in the field agree that by 2050, if we do nothing, the majority of coral reefs worldwide will be massively degraded. That is unheard of for a consensus like that in our community, because we're very contentious as a community, which is half the problem. Yes.
You said it earlier, I think, that the stressors are occurring faster than the ability for corals to adapt, and we can't control the stressors because it involves politics and people.
Yes.
So why don't we go the way of helping the corals to adapt faster?
Yes. That's really what we're trying to do, but we're trying to do not just one thing. So I am very much in the camp of it's not an either or scenario. We need to be doing absolutely everything in our wheelhouse to protect corals at this point. And the things like marine protected areas, for example, that's massively important to minimize the impacts on natural systems that can be controlled. Right? Marine protected areas essentially designate an area as a no-fish zone or a place where people will be extremely careful in their interaction with nature. Will that be enough? No, because we know there's just not enough time for corals to reproduce and replenish themselves with a number of excursions outside normal environmental ranges. Yeah,
On the other hand, on the plus side,
yeah.
in your research and other people, how many super species, super variants have been discovered
Yes.
that can possibly be used for regenerating reefs? Is it small? Is it spread over the different species?
Well, I would say that 50% of the world's reefs are supercorals at this point because they've survived very, very big disturbances in the reef. So we have a lot of material to work with. It is many different species, and within a given species, many different individuals within that species appear to be very strong. And that's actually what we're focused on here in Hawaii. We have relatively low diversity coral reefs. So we have 5 species that are quite dominant in Kaneohe Bay, for example. The rice coral, the finger coral, we have the huge mound massive, and then the lace coral, I think, and I'm trying to think of we call them Possumopera meandrina and Husselopera danicornis. They're beautiful. We found individuals with all of those species that are doing much better than expected. And we know that because there were 2 back-to-back bleaching events in Kaneohe Bay in 2014 and 2015. So we were lucky enough to have, well, I sent my team out, 22 people in the water during that bleaching event, and we tagged hundreds of corals during that event that we knew their response to the temperature disturbance. So we know exactly which corals did well and exactly which corals did poorly. And so we work with the corals that did the best. The goal is to take the best and make them better. So the ones that didn't do quite as well, we're not interested in selectively breeding them, but we have hundreds of colonies to work with, and we collaborate with others who are cryogenically suspending the gametes of corals. So taking the eggs and sperm from corals and putting them into deep storage, much like we do with human eggs and sperm and embryos.
That isn't solving your problem because you still have a great decline in the corals.
That's right.
You need to be implanting
Exactly.
these all over, whereas before you got breeding superwings, take what you have, chop them up.
That's right. So all of that is happening. All of that is happening. But if you do that randomly, the likelihood that the things that you're planting will die is very high. And in fact, we have a whole industry on restoration of corals that is really going out, finding things that grow fast, not that do well to the environment, cutting them into lots of pieces, growing a lot of the same individual, and planting those. And we've had big temperature disturbances that come through and wipe that out. So the goal is to combine the approaches, take the very best of the best, identify them, breed them, and then perpetuate them and propagate them in the way that you're talking. So the goal isn't to have one solution. The goal is to combine all these solutions together to effect the greatest possible outcome. The reality is none of these solutions will fix all problems on reefs globally. There will be some solutions that are better in some places. There will be a suite of other solutions that are better in other places. But I guess the take home for me is to do nothing at this point, which is actually, for the most part, where we are, is very, very dangerous. Yes?
Yes. Can you go back to the first slide? No.
It's just slow, getting there. There, no. Symbiodinium?
Yeah. It's right over down here.
Yeah. That's the microalgae. That's the actual formal Latin name for the group of tiny plants that live inside of the corals themselves. They're called Symbiodinium, and in fact, I said they were all thought to be in one species. That species name was Symbiodinium microadriaticum, which was the longest species name I'd ever heard of, and I used to hate having to say it. But now we don't have to, because we just call them Symbiodinium sp, because there's so many species.
Is that the same algae that lives in the giant
It
plant?
is the same algae, except in the giant plant, it lives outside of the cells, not inside the cells. Same algae. Yes. Yes.
In Fahrenheit, what is the temperature range where it's at?
That's a good question. I don't deal in Fahrenheit. I'll get back to you on that.
I'll look it up.
Yeah, no, I'll get back to you on that. Yeah. So they like to live between about 25 and 31 is the absolute maximum. 32 is almost lethal. So they're really, in some parts of the world, it is 28 and 31. And in other parts of the world, it is 29 to 33. So it depends on the conditions of the place. So the Red Sea, for example, is a great example of extremely warm corals. And people have said, well, should we take corals from the Red Sea and implant those in the Pacific? And the answer is, if you did that, they would die because they couldn't withstand the low end of the temperature range. So there's a lot of nuance in how far you can move a coral, because what they like is what they like. They're very finicky, I would say.
Corals, it's the symbiodinium that they're eating or using for nutrition.
They're using it for nutrition, but they don't eat them. They just live in harmony with them.
But then a polyp is eating little things in the water. So what else does a coral consume?
It's a very interesting question, actually, because several species are very active feeders. And often when you see very large corals, large corals with lots of tentacles out, that's a sign that they're active feeders. So they feed on zooplankton, tiny little things that swim past them in the water column. They have what are called stinging cells, nematocysts, on the end of their tentacles that literally trigger and shoot out a harpoon and catch something as it swims by and then pulls that back into its mouth. I mean, it's remarkable. But there are some corals that don't feed at all. So they are exclusively dependent on the algae inside of them. So this is what I mean about the complexity in the group. It's so large that one rule for one coral species will not fit another coral species because of differences in their basic biology, like their feeding or even their reproduction. You know, within the reproductive modes in corals, we have sequential hermaphrodites, we have single-sexed corals, we have brooders, things that produce live larvae that swim immediately around rather than having to be fertilized from an egg and sperm. We have things that just crack and fragment and will form a new colony, just like the gentleman was saying. You can cut them up and grow many different replicates of the same individual. Stick them on a reef. You can do that. It's being done all over the world as a response. Yes.
I want to hold my question until you're finished. I want to give you a chance to finish.
Well, I mean, I can finish because I don't want to keep you. What time is it?
We have 2 minutes.
I've got 2 minutes. You see, I was just going to cut me off. It's all right.
Can I ask my question?
Yes.
My question is probably a couple of parts. So let's assume you do research.
Yes.
And you find your super coral. And you got the properties that you want. And you find the symbiont that works very well with this. Where would you first want to locate your trial? I mean, you've done it in your lab.
Yeah.
Now you want to find a place to put it.
Yeah.
Where would you want to put the first trial?
It's a really great question. So I think the decision about where actually belongs to many more people than just me. So because we actually have to convene with people who study large-scale ecology. So for example, if I took an island, there are reefs on that island that we will call source reefs, that they produce a lot of the reproductive matter that then go around the island based on the actual flow around the island, and they replenish reefs around the island. So what I'd be looking for now is all the source reefs. I want to know where the source is because I want to actually increase the return on investment. I not only want to replenish the reef that I'm planting those supercorals on, but I want to invigorate the reefs that actually connects to and provides offspring to. So there's a lot of discussion about understanding with the oceanographers what the flows around the island are, which reefs actually are the source for propagules, reproductive propagules, and which the sink, which take them up. So, but there are also issues of what do the people who uses the reefs? How can we actually think about the reefs and replenish the ones that actually are going to be most important to servicing, say, the fishery for a given place? That has to be in our thought process.
Okay, now you got your ex-source reef.
Yes.
Or location. Do you have to, wherever you pick it, I don't know where it's gonna be, or wherever you find it, what's the legal problem?
Oh.
How would you get permission to make that first run?
So this is such an important set of discussions because how we move the needle from where we are now to where we go is very difficult, and it's a very multi-stakeholder endeavor. So half of it is, first of all, talking to all of our governance bodies. So Department of Aquatic Resources governs all sampling permits that we are actually given to even start to explore this work. What happens next is a discussion with the Department of Aquatic Resources about whether or not they will allow any of these genetically directed, not engineered, corals to go back onto the reef. Now, in other places, French Polynesia, for example, they have lost so many reefs already that their tolerance to do this is really, really high. It's not academic anymore about whether or not something's going to go wrong with a reef. They've already lost the majority of their reef. In Christmas Island last year, 95% of the reef died. And that community depends on its reef. They will do anything to get that reef back. Now, does that make me happy or sad? It makes me so sad that we are at this place, but the reality is the tolerance to try and solve the problem is high in places where the problem is most intense.
I'm sorry.
Now you don't have any problem, you got this thing in the water.
Yep.
How long do you need, or your colleague need, to observe what goes on? How many years do you need before you can come to a definite conclusion that it's successful?
The corals need to grow, they need to reproduce, and they need to attract other
How
organisms.
many years do you have?
So I would say 5 years, because they will be hit with a temperature disturbance in that period. To get through that will be approved. So it's, it's out there, but we have to do something. Thank you.